Next speaker is Tyrone Spady. And I would first of all like to thank Tyrone for agreeing to do this little switch to accommodate our third speaker, who has arrived, by the way. So we'll give him a minute to collect himself, and then he will go as the last speaker. Tyrone is M7, and he went on to get his PhD from the University of New Hampshire and in zoology. And he is currently with the Human Genome Research Institute at the NIH. And his talk is Canine Genomics, Mapping Behavioral Traits. Tyrone.
Okay, so my talk is actually going to be a little different from what you have been hearing so far. First of all, it's not a quantitative talk. What I'm going to do is focus on providing a framework for how we think about identifying the genes that underlie behavioral variation in purebred dogs. First of all, I'm going to define a few key terms, the first of those being a genetic variant. This is basically a polymorphism, a polymorphic segment of DNA of known location. So this will take you back to your first genetics class. You get one of your chromosomes from your father, one from your mother. So this genetic variant is a place where the maternal and paternal chromosomes are different. These are two common types of genetic variants, SNPs, or single nucleotide polymorphisms, which result in, which are single base differences in the DNA sequence, and also insertions and deletions, or indels, which are differences in the presence or absence of base sequence. So what we like to do is use these genetic variants to actually identify the genes underlying complex traits, or disease susceptibility, or in my case, behavior. So we use an approach called genome-wide association, which basically looks at the genotypes at cases and controls and looks for statistical association between markers distributed across the genome and a particular trait of interest. So here's sort of a simple example, a simplified example of what you like to see in one of these sorts of studies. So basically what you're looking for are parts of the genome that are shared by all of the individuals that share your trait, or cases that are excluded in individuals that don't have your trait, or the controls. So physically closely associated markers are typically inherited as a single unit. These units are called haplotypes, and this is because of a property called linkage disequilibrium. Oh, and the picture's gone. This is because of a property called linkage disequilibrium, which basically is the non-random segregation of alleles, and that basically has to do with, again, the closer two genetic variants are to one another, the more likely they are to be inherited together. So in the dog, linkage disequilibrium is really extensive, and that has to do a lot with the structure and the creation of dog breeds, because the breeds were typically started from relatively few numbers of individuals, and also popular sire effects can be really important in dogs. In other words, if a dog wins a major dog show, that dog is likely to contribute a lot to the next generation. So because dogs have more extensive linkage disequilibrium, they also have larger haplotypes. And basically what this translates to is that fewer markers are required for mapping. So in humans, these haplotype blocks are usually less than about 50,000 bases. In contrast, in dogs, these blocks can be larger than 2.5 million bases. So to do this type of whole genome association study in humans, they typically start out with about 500,000 markers, again, spread throughout the genome. And in dogs, we are estimating that you can do these types of studies with as little as 10,000 markers. So we're actually, we have a product, Canine SNP chip, that has 127,000 of these markers. And actually you lose a significant portion of those just because of they don't work, or the dogs aren't polymorphic at those particular loci. These 127,000 markers were discovered as part of the Dog Genome Sequencing Project, which was published in 2005. And they are among the 2005 SNPs that were discovered as part of that effort. So we have these wonderful genetic tools to be able to apply to the canine system, and the point and the purpose of all of this is to begin to understand some of the amazing phenotypic variation that we see across the more than 400 breeds and varieties of dogs that are recognized across the world. So breeds by definition are typically fixed for very distinct phenotypes, or they have very characteristic sets of traits. So, for example, the Scottish Terrier in the upper corner there, those dogs tend to all have, they have a mild form of achondroplasia. They usually have very similar types of quality of coat, as well as, this is kind of funny, their skeletal proportions are also sort of stereotyped. So dogs have kind of morphological traits that seem to be, that are fixed and breed defining. So in the same way that there are characteristic morphological traits, there are also characteristic behavioral phenotypes. And a lot of this has to do with the fact that breeds were developed to perform very specific types of jobs. So for example, we have breeds that were developed to chase and hunt game, breeds that were developed to point and flush game. You have the Labrador Retriever, which is a retrieving breed, the Border Collie, which was developed to herd, there's a livestock guarding dog at the bottom, the Swiss Mountain Dog as a drafting animal, and that's a little beagle, which was developed to track and trail game. And all of these specific jobs, this is crazy, all of these specific jobs have very characteristic suites of behaviors that have been optimized for those tasks. So they result in a lot of behavior variation across dog breeds today. So we have a number of tools to begin to capture some of that behavioral variation, and those being the expert rating of breed prototypes. This is probably what people initially started to do, where basically you rely on people who are extremely familiar with dog behavior, such as dog show judges or veterinarians or behaviorists to actually assess breed averages for behavior. And that's not so useful from a mapping perspective. The gold standard is the test battery and the observational assays. These document the behavior of individual dogs, but they represent sort of hands-on analyses of the dogs. So the difficulty there is that they're relatively time-consuming and extremely cost-prohibitive. So the method that our lab is actually focused on is to use informant-directed surveys to learn about individual dogs. And the neat thing about this approach is that it utilizes the expertise of individual dog owners to characterize the behaviors of dogs. So really briefly now, I'm going to kind of just give you a flavor of, I've given you kind of a conceptual framework of what we do in terms of trying to understand the genetic basis of complex behaviors. This is an example, and this is a case study in actually a Cocker Spaniel. So a couple purchased a male Cocker Spaniel puppy from a pet department of a well-known London store. The first weeks were uneventful, but by 10 weeks of age, it growled when the food bowl was removed. At 13 weeks of age, it severely bit the owner's hand when she attempted to repossess a bone, and other such incidents followed. At 7 months of age, there was a marked increase in the frequency of confrontations over stolen objects, and it growled when stroked in its bed. The dog was castrated at 8 months, but to no effect, and it continued to deteriorate. Eventually, the dog would go on to seriously bite the owners on at least 20 occasions, as well as visiting children twice. The dog never bit strangers outside the home, and the dog was later euthanized. And this is really a textbook example of this severe owner-directed aggression. And we're actually conducting a study right now in a closely related breed, the English Springer Spaniel, to begin to map the genes that are controlling this trait. And there are a few characteristic things about this case study that I'm going to point out. One, that the aggression is primarily directed towards owners, and the other is the relatively early onset of the aggressive behavior. It typically occurs before social maturity. So in the English Springer Spaniels, this is the breed that we most commonly see displaying this aberrant aggressive behavior in the U.S. It's the breed that's most likely to bite the owner. In a 2005 survey of English Springer Spaniel owners, over 2,000 surveys were sent out to English Springer Spaniel owners. About 1,000 people responded, and they indicated that about 26% of English Springer Spaniels had bitten people. And family members were usually the targets of that aggression. And when we actually looked back at the pedigrees of those individuals, so looked at the genetic histories of those individuals, we were able to identify one sire and one particular kennel that were overrepresented among the aggressive dogs. So that's sort of strong evidence that this is a tractable behavior with genetic basis. So I can't really get into the data too much, so stay tuned for that. And so really we're at a wonderful time in terms of canine genomics, and that with the recent sequencing of the canine genome, we have wonderful tools to begin to understand the genes underlying the behavioral diversity that we see in purebred dogs. And ultimately, we want to realize the promise of the Dog Genome Project, which in part was to understand the genetic vocabulary of behavior and also disease susceptibility. So in the future, we'll probably begin thinking about behaviors such as fear, sociability, playfulness, as well as the whole suites of behaviors that are associated with those specific tasks that dogs have been bred to do. Really briefly, I want to thank dog owners for the generous donation of samples for our studies, as well as Elaine Ostrander, my postdoctoral advisor in the Ostrander Lab, and the Meyerhoff program for supporting me through undergrad and also inviting me to be here. Thank you.
We have time for brief 1 or 2 questions.
Okay.
You said stay tuned. Do you want to give us a little bit more on what you have in mind for stay tuned?
We actually have a candidate loci for the aggression phenotype. That's about all I'm going to say. Yeah, so actually understanding the breed relationships is a really important part of what we do because we use that information to sort of in the design of mapping studies, with each breed sort of representing an independent, you can think of them as an independent experiment. So if 2 breeds or mixed breeds share a particular phenotype, then it actually makes it easier for us to find the genetic variant. So I said that the study is primarily being done in English Springer Spaniels, but we're also looking in closely related breeds that also exhibit the phenotype. So the English Cocker Spaniel is the other one.
So for humans, we say nurture and nature play a role, right, in the development of a human being.
Yes.
For animals, for dogs, nurturing doesn't play a role?
No, there's probably tremendous environmental variation in the trait, and that in part is the difficulty of looking at any kind of complex behavior, and that's a difficulty that we have with the phenotype. For our mapping study, we actually have 192 dogs, and we were only able to use we chose to only use the 60, which represent the extremes of the behavior phenotype. So we chose dogs that had bitten, usually dogs that had bitten multiple times, and compared those to dogs that had never shown any kinds of aggression. So, I mean, even with that kind of dichotomy, there's likely dogs that are biting because of environmental reasons, because of experiences that they've had that have sort of allowed aggressive behavior to come to the surface.
But you're focusing on the genome side.
Yes. Well, ultimately, the genes will represent the potential to express any kind of behavior. So I don't think that it necessarily has to be all of the other. But I think there's definitely, I mean, the fact that this particular phenotype segregates within a breed strongly suggests that there's a genetic component to it, is the basic. Yeah. All right.